In this video, we will discuss about this two-step CAR expression on T cells. This is the idea that can be used to reduce immunogenic effect or immunogenic reactions in the patient, as CAR will express only when it interacts or when it is able to see the target cells. Here it is represented here in this figure as signaling cells. So what is this idea? Is that the signaling cells are present, and instead of engineering T cells in a way that will express CAR before it is transduced into the patient, it will engineer in a way that it will express these notch receptors that can see the signals or that can interact with the antigens that are present on the tumors. Once it interacts with the signaling cells, it will trigger this syn notch receptors, which is based on the transcription factors, not a cell signaling activation pathway. So these transcription factors will then move into the nucleus or translocate into the nucleus, where it will encode the CAR gene, or CAR encoding gene will start to express once this transcription factor translocated into the nucleus. And thereafter, once the CAR is expressed, it will then translocate it into the cytoplasm and then to the cell surface. So this is a 5-step process. So in this case, the CAR will express only when these notch receptors extracellular membrane domain interacts with the signaling cells or the target cells. So this will make the T cells more specific to express CAR in the vicinity of the target cells. This will reduce the immunogenic effect at other sites. So one of the problems that might arise in this approach is the trafficking of the CAR to express on the cell surface, because these transcription factors, once it is inside the translocated into the nucleus, then it will starts to express or encode this CAR at a high rate, and this will cause a high expressions or more trafficking of the CAR expressions, and that will cause overexpressions of this CAR on the engineered T cells. So this is one of the problems that people are speculating in this kind of genetic modifications or engineered T cells. And one more things that this researchers presented along with this figure is to control this GVHD, that is graft versus host disease. In that case, what they are suggesting is to engineer T cells in a way, for example, it will become sensitive to a small molecules. And the examples that the researchers provided is about the inducible Cas9. So this inducible Cas9 is by nature inactive in these genetically modified cells, genetically engineered T cells. But once a small molecule inhibitors injected or introduced into the patient, then it will interact or activate this inducible Cas9, which is not shown in this figure. So it will activate this inducible Cas9, and that will trigger the rapid apoptotic cell death. So in the case where people are seeing the more immunogenic response in the patient, this kind of switch kill can be used if it is engineered into the T cells, so that the immunogenic response can be suppressed immediately or within a short period of time. So this is 2 approaches that are mentioned. The figure of the one approach is shown here, and for the other approach they have mentioned it only into the text. And they have also provided evidence or data in order to support their second idea, where they have provided a reference in which they shown that this iCasp9 has been used in a patient who has undergone allogeneic lymphocyte depletions, and thereafter the stem cell transplantations and seeing the GVHD symptoms. The small molecule inhibitors is being used to kill these genetically engineered T cells. So in this patient, the T cells that are engineered T cells that are transduced are modified with this iCasP9. So that is one of the reason why they are able to use this switch kill option by inducing this small molecule inhibitors that can activate this iCasP9 and trigger rapid cell death of the genetically engineered T cells. So these two approaches can be combined to reduce the immunogenic response, at the same time increase the CAR efficiencies to the target cells, against the target cells, CAR efficiencies and CAR anti-tumor efficacies against the target cells. That's all for this video. If you need to understand more about this idea in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.